All right, so I hope everybody's doing well. We're going to get started here on tissues. I want to emphasize to everyone that this is usually for the college students and high school students one of the hardest sections, just because there are so many in number. It's one reason why I've broken this down into small chunks. So, first, as the basic outline shows, we're going to go through basic definitions of tissues. We'll look at their cellular connections and how the cells are held together. We'll also then start breaking the tissues up into their individual types. After that, we will take a look at the different membranes inside of the body. And the last piece is we'll look at tissue repair. So the first thing that we look at with tissues is their ability to hold each other together. And so we look at tissues as being that group of cells that have a basic similar structure. And because they have a similar structure, that they usually perform some type of common or related function. And because of this, we look at the way those groups of similar cells are held together. And we have three basic types with one subset. The three basic types are tight junctions, desmosomes, and gap junctions. You'll notice on the slide that we throw under desmosomes what are called hemidesmosomes. All four of these help connect one cell to another. And we're going to talk about how those connections occur. So first, let's talk about tight junctions. Tight junctions are going to be found between cells, and as their name implies, they hold very tightly together, and they are so strong that they not only prevent the cells from tearing apart easily, but they prevent any movement between the cells. They do not allow water or nutrients to pass between the cell membranes between these two cells. Desmosomes, not as strong as tight junctions. Desmosomes will be found between cells, holding cells together. And these act almost as like rebar when we're talking about building a house, is they help reinforce the junction, the ability of holding these two cells together. Desmosomes are not as strong as tight junctions. Because they're not as strong, they do allow water movement around them into the cells, but they prevent most nutrient movement between the cells. Underneath desmosomes, you'll see the subset that is called a hemidesmosome. Hemidesmosomes are found only between the cell and the basement membrane, where the cell attaches to the underlying connective tissue that holds that grouping of cells together. When we get to the pictures, we'll really emphasize the difference between desmosomes and hemidesmosomes. The last one on here is gap junctions. And gap junctions are where two proteins from two cells line up and create a tunnel from one cell to another. By creating this tunnel, it allows for movement of nutrients and water from one cell to another based on the concentration gradient. Remember from the last chapter, we talked about in a concentration gradient, things move normally from high to low concentration. Because of this, if one cell has more of a nutrient or more water, we can move that excess nutrient or water through a gap junction towards another cell that has less. So in this slide, we see that to the left, we have two large cells that are side by side and the wall of a third cell that's attached. And we can see the three types of junctions located in this wall. Towards the top of the cell where we have the microvilli, we see tight junctions. Below that, towards the middle of the two individual plasma membranes, we see what are the desmosomes. Towards the bottom of the cell, where it's attached to the underlying basement membrane at the very bottom, we see what are called gap junctions. What we don't see here is an attachment at the bottom of the cell to the basement membrane. That's where we would find the subset of desmosomes that are called hemidesmosomes. Remember, all of these three types of junctions, tight junctions, desmosomes, gap junctions, and hemidesmosomes underneath the desmosomes, are all there to help hold cells together. Tight junctions, really tight interlocking proteins that prevent movement of water and nutrients. Desmosomes, not as strong, made up of those intermediate filaments that we had talked about last chapter, and those intermediate filaments create this reinforcement of the attachment. And then towards the bottom of the cell in this picture, we see the channels. Remember, channel or pore proteins. The channels that attach from one cell to another and create the tunnel that allows nutrients to move easily from one cell to another. Now remember these different junctions as we move forward through tissues, because we'll talk about how tightly some of these individual cells and tissues are bound together. So when we move into the actual discussion of tissues, we have a vocabulary piece that I want to make sure that everybody understands. We will talk about the four main tissue types. In many of the questions when we get to quizzes and exams, I'm going to ask you to name the tissue type, and then I'm going to ask you to name the tissue. The tissue type is broken into four groups. Every single grouping of cells and tissues, cells grouped together as a tissue in your body, is in one of these four groups. Either we are looking at epithelial tissue, connective tissue, muscular tissue, or nervous tissue. Now, in general, epithelial tissue covers and is there for some type of protection as a covering. So we look at your integumentary system, your skin, as epithelial tissue. But we also look at that anything that would normally have access to the outside world is also covered by epithelial tissue. So we look at what is going in through your mouth, the esophagus, down into your stomach, small intestine, large intestine, to the rectum, and then back out through the anus. All of that has some level of epithelial covering. The second grouping of tissue that we look at is connective tissue. With connective tissue, by definition, it tells you what it does. It connects one part of the body to another. It's there to help support, it's there to transport, and it's there as storage. But its main function is to connect one part of the body to another. One of the parts that students normally struggle with connective tissue is that there is a wide range in its makeup or how we see it. Connective tissues can either be a solid, they can be a gelatinous form, and they can even be a fluid. So as we go forward with this, that's one of the pieces that you need to pay attention to, is what does the structure actually appear as. The third tissue type that we look at is muscle. And remember, we've talked about this before, that we have three types of muscle in the body. We have what is skeletal muscle, we have cardiac muscle, and we have smooth muscle. All three types of muscle have one main function. That one main function is to cause movement. So, skeletal muscle helps us with the bones to cause movement of our body. Cardiac muscle helps us with the movement of blood through the body. Smooth muscle helps us with movement through very small tubes. Think blood vessels and digestive system. The fourth type of tissue that we look at is called nervous tissue. And nervous tissue main function is to control. We look at that it takes all impulses coming into the body, all stimuli, and it helps us with all output. It controls what we do. All right, the first grouping of tissues that we're going to look at is epithelial tissue. And you'll notice on this slide that we have multiple types of epithelial tissue. We have simple squamous epithelium. We have stratified squamous epithelium. We have simple cuboidal epithelium, stratified cuboidal epithelium. We have simple columnar epithelium, stratified columnar epithelium. We have pseudostratified columnar epithelium, and we have transitional epithelium, giving us 8 different types of epithelial tissue that we have to know by its name, its appearance, where it's located in the body, and what it does. The last piece that we look on this slide talks about glandular epithelium, the glands of your body. And we're going to talk about the different types of glands, and that those glands are made up of epithelial tissue that create the secretions that are circulating through the body, or secretions that are secreted out of the body. All right, so when we look at epithelial tissue, we do have some definitions. So the next couple slides here are just looking at definitions. The first one is, what is epithelial tissue? I've talked about a little bit that it covers the surfaces of the body, or it lines cavities in the body that have access to the outside world. It also lines inside of cavities that stay internal. So what we talked about before as around the peritoneum. Also, when we look at the pleura, we look at what is the pericardium. Those individual cavities have epithelial lining. So we see here that epithelial has 4 main functions. First, that it's there for protection. It adds an actual line of protection based on the number of cells that we see. The more cells in the tissue, the more protective layer it is. We also look at that it helps to control permeability, or movement in and out of the cell. Remember that the plasma membrane helps with that movement of fluids and particulates in and out of the cell. We see the same thing with epithelial tissue. The thinner the epithelial tissue is, the more likelihood that we're having movement of particulates. The thicker it is, the less likely. We also see that epithelial tissue helps with providing basic sensation. It's where we see a lot of the nerve endings attach within epithelial tissue so that we can receive that sensation. And the last piece of this is that it helps to produce what are the special secretions. And we look at what is going to be the epithelial tissue that helps with glandular productions. So sweat, saliva, hormones, all of that is going to be produced from specialized secretions from epithelial tissue. The next part we're going to talk about actually works with the picture that's on the next slide. And so we'll bounce back and forth between these two slides. First, we look at some pure definitions as cellularity and polarity. So cellularity looks at how closely that these cells are tightly packed together, or are they loosely arranged. If we have groupings of cells that are really, really tightly packed together, lots of tight junctions, then we say that the tissue has a high level of cellularity. If there's not lots of tight junctions, and that the cells have more space between them, we say that there is a low level of cellularity. So cellularity just looks at how tight these groupings of tissues are. Polarity is not a positive or negative. In this case, polarity is looking at the surface of the cells. So if we're looking at the surface that is attached to the basement membrane or the basal layer, that is going to be the basal surface. If that grouping of cells has a surface that's not attached to anything, then we call that the apical surface. And I will show you those examples on the next slide. So when we look at those attachments, we talked about that we have tight junctions, that we have desmosomes, we have gap junctions, and then we have the attachments that are attached to the bottom layer, the basal layer, and those were the hemidesmosomes. At that attachment to the basal layer, you guys are going to see many different names here. So we have the basal lamina, we have a basement membrane. Those are talking about the attachments from the cell to its underlying tissue. And that next layer of tissue is usually, if not always, some type of connective tissue. The next term that we look at with epithelial tissue accounts for all of the epithelial tissues. They are avascular, meaning they have no blood supply. For epithelial tissue to receive nutrients, it's only done through diffusion. So we have to have blood vessels that release nutrients around the tissue, and then the nutrients diffuse into and from cell to cell by diffusion. The last part that we look at with epithelial tissue is regeneration. And most epithelial tissue in your body has a very, very high rapid cellular division, meaning that they will replicate quickly. The only thing I would say with regeneration is that we have not destroyed the cells completely. If we destroy the cells, they do not have the ability to replicate. All right, so with this picture that we see of a group of epithelial cells, you'll notice towards the top of the picture that it has cilia and microvilli, that along the edge of the cells, it shows you that there towards the top are tight junctions, there are desmosomes, and then gap junctions towards the bottom, and that we are attached to a basal lamina, that individual grouping that's at the very bottom of the cell. So if we talk about this grouping that we're looking at, this would be a high level of cellularity. These cells are tightly packed together. That towards the top where we find the cilia and the microvilli, that this is the apical surface, the free surface that's not attached. Remember, microvilli are for absorption and secretion. Cilia are for movement of things outside of the cell. And then at the bottom of the cell of each of these groupings, where we have the basal lamina, that is the basal surface. That's where we would find the hemidesmosomes attaching the cells to the underlying basal lamina. All right, so the last slide that we're going to talk about here with this is the naming of epithelial tissue. So when we look at epithelial tissue, we name them based on a couple different characteristics or classifications. The first one is by how many layers of cells that we see. If we're looking at a single line of cells, then we call that grouping simple. If we see many layers of cells stacked one on top of each other and they're all predominantly the same type, we call that grouping stratified. Single layer, simple. Multiple layers, stratified. The third grouping of name that we have here is pseudo-stratified. Now remember, pseudo means fake. Remember that in this case, we're looking at pseudo-stratified, so it's fake stratified. Meaning that it appears as it being multiple layers, but it really is only a simple layer or a single layer. The problem is, because it looks like it's multiple layers, we cannot call it simple because there is a small change in this. So pseudo-stratified, all of the cells attach to the basal layer, to the bottom, to that basal lamina. But not all of the cells attach to the apical surface. This is a very small but definite change here. Pseudo-stratified, all the cells attach to the basal lamina. Not all of the cells attach to the apical surface. This is why it looks like multiple layers. The second way that we classify is by what we see, or their shapes. So we also will say that we have squamish cells or squamish cells. These are usually fairly flat, and I reference them in class as looking like flat fried eggs, in that no two fried eggs ever look the same. They have irregular shape, but all of them are flat except for the place where we have the yolk. For a cell, that's the place that we find the nucleus. So squamish cells are, to me, similar to flat fried eggs. The second grouping that we look at is cuboidal. Cuboid, think cube. So we end up with a cube-shaped cell, roughly the same height and width and length of a cell. Now I will tell you, the one thing with cuboidal cells is sometimes they appear almost perfectly round. So think cube in shape, but they can have soft or rounded surfaces. The third grouping is columnar. And columnar sounds like a column, meaning that it's taller than it is wide. So columnar cells will be very, very long, but not very thick in shape. And the last grouping that we have by its shape are called transitional cells. And transitional cells are exactly that. They're in a transition. They make up none of the other groupings. They're not flat fried eggs, they're not cuboidal, they're not columnar cells. They can be made up of any other varying shapes, and their main function is to allow to change their shape in that they can stretch.